Today we're going to hear from our very own Dr. Nuria Carrillo. I am so proud to announce that earlier this year, as you guys know, this month, September 1st, she joined us officially at the Neuromuscular Disease Foundation as our very own Chief Medical Officer. And we are really, really lucky to have her. As you guys know, she is among the world's foremost experts on GNE myopathy. As a physician and a principal investigator at the NIH, she's devoted the last 10 years of her life primarily to GNE myopathy and has multiple peer-reviewed publications that really are very, very impressive. Please check out her bio on our website at curehibm.org. And today we're going to hear from her how her inclusion on our team is going to help us expedite our journey to gene therapy. And yes, it is a journey. So hopefully at the end of this, you guys will have a bunch of questions, and you can please post those in the chat box as they occur to you, and I will sort through them. And I'm going to ask everybody to please keep your cameras and your mics off. But without further ado, I'd like to invite Nuria to take over and share her slides with you. Good morning, Nuria.
Good morning. Thank you for the introduction. I'm so happy to be giving this presentation today as part of the last one of this GNEM Symposium Speaker Series for 2020. I am going to, okay, there we go. As part of my talk, I will identify one of my goals as Chief Medical Officer, which is to expedite genetic therapies for GNE myopathy. And as part of this, I will give an overview of the scientific advances that have happened since the discovery of the gene in 2001. So that's 2 decades ago. And also some of the current research programs that have highlighted the specific things that are needed for a successful gene therapy program. And I will, as part of this process, help us identify further research that may be needed to advance gene therapy for clinical trials. I am going to start the presentation with the history of how scientists have discovered what we know about GNE myopathy. So why is this important? First, because we can learn much from history, and I like history, and also because 2021 marks 2 decades since Dr. Stella Mitrani-Rosenbaum discovered the gene for GNE myopathy. And so we think it will be befitting to commemorate 2021 in her honor, 2 decades after the discovery of the gene. And as you will see as part of my presentation, the majority of the advances in the field happened starting when the gene was discovered. And so I would like to show an overview of the timeline from the moment the disease was discovered in the 1980s and first described clinically and muscle biopsies, to the search for the gene in the 1990s, the identification of the gene in early 2000s that led to mouse model development, preclinical studies, understanding the pathophysiology of the disease, identifying patients and different mutations in the gene that lead to GNE myopathy. In 2010 to 2020, we have seen the majority of the clinical studies that have happened for GNE myopathy, and we hope that starting in this decade, that we will see an advance towards genetic therapies for the disease. So let me start with what I think may be the first description of GNE myopathy, which was done by Dr. Nonaka in Japan in 1981. He described three cases from 2 families of patients with muscle weakness affecting the anterior tibialis that had onset in their 20s, mildly elevated CPK, and muscle biopsies that showed green vacuoles, no inflammation and no regeneration. He also noticed that there was autophagic activity, vacuoles, This is the apoptosis. This is the natural mechanism by which cells program their own death. And it is interesting that in just this single description from the 1980s, the majority of the clinical and histology features that we now know were described in a single paper. So we owe a lot to Dr. Nonaka and his very detailed and thorough description. In 1984, oh, and by the way, Dr. Nonaka named the disease back then distal myopathy with rim vacuoles, and some people also refer to it as Nonaka myopathy. On a separate investigation in 1984, Dr. Argov described nine cases from Iranian Jewish families that had a very characteristic autosomal recessive inheritance with onset in young adults. And as we know, the sparing of the quadriceps was described during this paper. At that time, the disease was called hereditary inclusion body myopathy, or HIBM. And so in the 1990s, since the disease was described, there was an effort led by Dr. Mitrani Rosenbaum in Israel to find the gene. In 1996, her group mapped the location of the gene responsible for what they known back there as HIBM to the short arm of chromosome 9, and you can see this on the slide. And then in 2001, in this wonderful paper in Nature Genetics, her team described the identification of GNE as the gene responsible for HIBM. And interestingly, just a year later, in 2002, Dr. Nishino, who worked with Dr. Nonaka, described that the same gene was affected in Japanese patients with distal myopathy with rim vacuoles. So we have different names from the disease, but one gene, one condition. In the later years, so when it initially was described, there were patients from different regions, mostly in Israel and India, that were described on the original paper by Dr. Mitrani Rosenbaum. The next year, we saw then patients in Japan having GNE myopathy. And over the next several years, many more people worldwide started to be identified with patients with muscle weakness with mutations in the GNE gene. Over time, we knew that we have identified more than 200 different mutations, and we currently estimate the prevalence of the disease to be roughly 6 in a million people. One of the issues in these years, in the 1990s and 2000s, was that patients had a very difficult time finding a diagnosis. We estimate that it took patients an average of 10 to 15 years to reach a diagnosis. They will go to a variety of doctors, and they were all very confused about what the potential diagnosis may be. Even now, we know that we estimate that there are 30,000 people with the disease currently undiagnosed. And so this is one of the reasons that back in 2013, a consortium of scientists working on GNE myopathy decided to unify the names HIBM, Nonaka, inclusion body myopathy type 2, and hereditary inclusion body myopathy to one name, GNE myopathy, to facilitate the diagnosis of patients with the disease. And as you can see here, Dr. Nishino is right there, and I am luckily in between Dr. Nishino and Dr. Mitrani Rosenbaum and several other people that you probably already know, including Dr. Argo, Dr. Kakis, and Caroline back then. So let me talk about why preclinical testing is important for developing gene therapy for GNE myopathy, and what are the elements that we must have to undergo the regulatory process that will allow us to get to clinical trials for GNE myopathy. And first, I am going to talk about mouse models, because mouse models is one of the tools that scientists have to be able to test different therapies before they move into clinical trials. And although we are not mice and we cannot always generalize the results of mouse studies to humans, they do provide a very powerful tool to test different things, including safety and efficacy before a therapy is tested in humans. So what are the characteristics of a good mouse model? First, that it is what we call biologically relevant, meaning that it represents the human disease. Next, that it's amenable to perform assessments. So if you need to know whether the gene therapy is improving muscle weakness and survival, you need to be able to measure these things on this mouse model. And also that it is reproducible, meaning that the mouse will show the same characteristics in one lab and another. So it could be used by different investigators while performing different evaluations. And so I'm going to give you an overview of the mouse models that we currently have for GNE myopathy. The first mouse model, and you may not be aware of this one, was done right after the gene discovery in 2002 by a group in Germany. And this mouse, as you can see on the screen, a piece of the DNA is broken off. This mouse, what they did was inactivate the whole epimerase function of the gene. And they found that these mice did not survive in utero. In fact, they die 8.5 days after conception. So therefore, this model, while very reproducible in many laboratories, it is not really biologically relevant, as it doesn't show what we see in patients with GNE myopathy. And there's no way to evaluate efficacy because the mice die before they're born. So we could not use this mouse model when it comes to testing gene therapy. The second mouse model that I'm going to talk about was done in 2007. This is what we call the Japanese mouse model. It was done by Dr. Nishino's group. This is a transgenic mouse model that is overexpressing the D207 mutation, which was previously known as D176V. And while this model shows the mice have muscle weakness starting around 20 weeks of age, and therefore recapitulates the human phenotype, meaning it will be biologically relevant because it is very close to what the disease is in humans. We have unfortunately heard that it has not been able to be reproduced in other laboratories. And the third mouse model was the one done in the US in a collaboration between the NIH and Dr. Darvish. And in these mouse, there we have the N743T, the Persian Jewish mutation. This model is not biologically relevant because it has kidney disease instead of muscle disease. So when I say we can perform functional assessments in mice, what do I mean? I mean that we can do a treadmill test, like the mouse model version of the 6-minute walk test. And the mice walk on this treadmill, and it's connected to a computer, and it measures the speed at what and the length and the amount of time, the distance that the mice can travel. And so this could be done in mice as a measure of improvement. And then there are even measures of grip strength. Here you can see the mice holding onto the grid, and on that machine, the grip is being measured. So there are a lot of ways in which we can assess efficacy potentially in a mouse model. And all of these are, there are protocols that have already been described to do these evaluations. So going back to the Japanese mouse model that developed weakness at 20 weeks of age, they used this preclinical model to test both sialic acid and MANAC. And in a paper by May Maligdan and Dr. Noguchi, Nonaka, and Nishino as the senior author, they actually were able to test a variety of things, including the distance that the mice were able to run. On the bottom right, you can see that L stands for the unaffected mice, the C stands for affected mice, and then the next 3 bars correspond to MANAC, then sialic acid, and then sialyl lactose. And they tested the ability of the mice that were treated with those to be able and show that they could run, they improve compared to those that weren't affected, and got to close levels to those that weren't affected. So in fact, this mouse model was used to provide the proof of concept evidence for the clinical trials that happened in the 2010 decade. Unfortunately, as I mentioned, the phenotype of these mice has not been able to be replicated in other laboratories. And there are many reasons for this. Factors that may impact a phenotype include a different genetic background of the mice, or accidental genetic contamination, what we call genetic drift, which is the inclusion of new mutations that happen naturally over time that may affect the genotype, and also the state of the mice health and infections and diets. So do we need a new mouse model? I think that we probably do. We either need to identify and fix the issues with replicating the Japanese mouse model, or we need to generate a new one. The good news is that since 2007 and 2020, there have been incredible technological advances in our ability to generate mouse models. So this is what we called a CRE-LOX-P system. And I wanted to show this to you because this is one of the most powerful tools currently in use by mouse geneticists, and probably the one that could help us yield a mouse model in GNE myopathy that has all the necessary characteristics that we want to be biologically relevant, to live long enough for us to perform evaluations, and for it to be reproducible in all laboratories. And the way these work is they use this technology to cut off, that they may cut off the GNE gene or sections of the gene. And they have a switch, literally an on and off switch, that gives the scientist the power to turn on or off a gene at the time that they want and in the tissues that they want. So that's what this is called, a tissue-specific and inducible mouse model. So they can induce the mouse to develop at any given time that they want this loss of expression of the gene of interest in whatever tissue they want. And not only that, but there are ways in which you can build this system that allows for a color marker to be expressed by the tissues and the cells in which whatever you want it to happen. So if you wanted to turn off GNE in skeletal muscle at week 20 and then see what happened, you would see the muscles at week 20 express this green signal that you can see under the microscope. And so it's a very powerful tool, and one that in her presentation back in June, Dr. Mitrani Rosenbaum discussed in detail. So if you're interested in learning more about this, please go to her presentation. I believe she talks about this starting in minute 15 of her presentation. And she discussed in detail her specific strategy to generate a new mouse model for GNE myopathy using this system. And so now that I have explained mouse models, I am going to move on to different types of genetic therapy strategies. They include gene delivery by a viral vector that was discussed in detail by Dr. Martin on September 15th, and I do recommend you go and see that video. And also what we call gene editing that was discussed by Dr. Monko-Lek and Angela Lek. And here I am just going to show you a small summary of the differences between these two genetic therapy strategies. When you do gene delivery by a viral vector, and this is showing a cell that has, as you can see in yellow, a change in the DNA, in this case, the GNE gene. And what the therapy, when you do viral vector gene delivery, you are in fact delivering a vector, which is a virus, which you can see here as a rhomboid, with an intact gene inside that then can go inside of the cells and release a copy of the unaffected gene that will be used by the cell's machinery to create a message that will create a protein that is completely functional. An important part of this strategy is how much to give. And that's why Dr. Martin is developing a potency assay so that it can guide the dosing for gene therapy. The other strategy, which is the gene editing, is you have literally a spelling mistake in the gene of interest. And what this strategy does is it will remove that spelling mistake and replaces it with the appropriate spelling. And that's what gene editing does. And these, as we know, there are more than 200 different mutations in the GNE gene. So this is specific to one. So for example, these would be potentially feasible for patients that are homozygous for founder mutations. But we know that 2/3 of the patients have different mutations in each one of their genes. So while these may be feasible for those that are homozygous for common mutations, it would be it will be harder to do for patients with less common mutations in the gene. And so before attempting clinical trials in humans, it is important that researchers show that preclinical testing, that means testing in models of the disease, show without a question efficacy and safety before proceeding with what we call a first-in-human trial. So this includes, obviously, the choice of genetic therapy strategy, whether it's going to be gene editing or gene therapy by delivery by an AAV vector. It also includes a very careful choice of the vector. What we call the vector is the type of virus where the gene is packaged, and the promoter to target the tissue. The promoter is a switch that tells the gene they should be expressed. And all genes have a promoter that basically turns on the expression of the gene. And so typically, for example, you have seen that for the microdystrophin strategy that they use for Duchenne, they use the AAV RH74 vector. That's because that has been shown to be useful to target muscle. And the MCK promoter, for example, that stands for muscle creatine kinase promoter. And after those, after that promoter, you can attach any gene you want. And the choice of this vector and promoter is primarily to target the tissue of interest that you need to correct with the gene therapy. And therefore, maybe obvious choices like this vector and promoter that has been previously used for muscle diseases may work for GNE myopathy, but you don't know until you actually test it. And that's one of the reasons we need a mouse model to be able to test that. And you also need to be able to show what the FDA calls proof of concept. And that's evaluate whether your strategy is useful at correcting not just the gene defect, but the muscle weakness. And mouse models also help us establish what's called a pharmacological effective dose. And whether that should be IV or IM, that's something that you determine in a preclinical model. What are the dose levels that are both effective and safe? Because you need a dose to identify a dose level that is both. And also whether one dosing is sufficient or multiple doses may be needed to maintain effect. And if so, how often? So really what the preclinical testing does is builds a strong foundation for a successful drug development program. If these foundations are not strong, the program will not be as strong as we need. This is the most important part of the process. And as I mentioned, one of these important tools, the development of the potency assays, are being developed by Dr. Martin with funding from NDF, because this will allow us, once we have a mouse model where we could test the efficacy, it would allow us to identify the dose levels, what we were talking about a moment ago. In terms of evaluating efficacy, so let's go through the process. You have a mouse that you successfully developed that is that has a change in the GNE gene that is biologically relevant, that shows the disease, that survives enough for you to test him, that shows muscle weakness. And so you decide to go ahead and give gene therapy to these mice. And so here we give an injection to the mouse with the gene therapy. The gene is a DNA. So once inside of the cell, you need to show that that DNA is being successfully made into what we call a message, which is the RNA. We have very good ways of measuring RNA production that have been used by scientists in the GNE myopathy community right now. So that is not an issue. However, that RNA gives rise to the protein. The protein is what carries out the function. When people talk about the need for antibodies, they are talking about you need an antibody that detects the presence of the protein, meaning that the gene has successfully created a protein. And here you can see how that's done. Plus plus means that there are no changes. Sorry about that. There are no changes. This is a mouse that has no mutations in the gene and therefore produces the protein well. This is a mouse that does not produce any protein because it has two changes, and this is a treated mouse where you could see some change. And there are antibodies that have been used over the years, but unfortunately they're difficult to use and not always show great measurement. And so this is something that should probably be improved to be able to detect that the protein has been made after giving gene therapy. But then after the protein, here you see the crystal structure of the enzyme, of the GNE enzyme. That it's a three-dimensional protein that acts to produce, and here we show the pathway, UDP-GLUGNAC into MANAC and then into MANAC 6-phosphate. And so one strategy would be to evaluate the protein function would be to measure the enzyme activity. And although there are several enzyme assays that have been developed, again, we need to do procedures to make sure that these are reproducible across all different laboratories, so they can be used by all scientists. And then the next level is, as you know, after you restore the enzyme activity, you restore the sialic acid pathway, the effects at the cell level and at the tissue level can be measured with what we call lectins. Lectins that identify the now sialylated glycoprotein in the surface of the skeletal muscle. Some of these studies have been done and were published 2 years ago at the tissue level, but Dr. Kelly-Crow is developing a lectin method that will allow scientists to identify these sialylated proteins that are kind of the ultimate tissue and cell level measure that the process has been restored inside the cell to be able to evaluate whether that is present in cells and mice that are treated with gene therapy. And so I'm just showing you again the DNA level, then the protein, the function of that protein, which is the enzymatic activity, the restoration of the pathway, which you can measure sialic acid levels, and then ultimately that sialic acid is attached to proteins that become part of the surface of the muscle cells, and that's what it's measured by these lectin biomarkers. But then the final proof that this is working, obviously, is in the mice to perform the functional testing to compare those treated and untreated mice and show that, for example, with grip strength or ability to run, that there is an improvement, a significant improvement compared to those untreated mice, and that should be close to those unaffected mice. And so this is what establishes the proof of concept for a preclinical program. And these are all important issues to think about. And once all of these have been solved and the ideal gene delivery construct, so the vector with the promoter and the route and dose of administration, and you have shown in this back and forth process until you find the ideal combination, then you decide to move that forward potentially into clinical trials. But before doing that, typically it is required by the regulatory agencies that there are toxicology studies performed. So what is the reason for toxicology studies? These are studies that are typically done in healthy mice. It could be done also in affected mice, but it's not necessary. And the main purposes are to identify an initial safe dose, to identify what your dose escalation may be. So here on the graph, you're showing at a certain dose, you start to have a toxic effect. And so you want to make sure that your therapeutic dose was shown to be effective in preclinical studies, but that less than 10% of the mice or the rats or the dogs that you're testing are showing a toxic effect, and none of them have any deaths from this dose. So that's how you identify what we call the therapeutic range, and you would start with the lower level of that range as your initial dose, and you will escalate the dose in that green window, but never going beyond the end of the green window so you don't reach a lethal effect. So what's called an initial starting safe dose and the maximum dose. It can also help you doing a toxicology study to identify whether there's going to be a host immune reaction to the vector and how intense this reaction might be, although you may not really know until you start testing patients. And it also helps you identify what sort of safety evaluations you will need to perform in the patients when the clinical trial starts. Toxicology studies are typically very expensive, but a necessary part of reaching clinical development. And so let's talk a little bit about clinical trials to end with. And as you know, there had been a handful of clinical trials. The first one in 2006, testing IVIG, which is a highly sialylated version of a plasma protein. This was a small trial with 4 patients and 1 month follow-up. Then there was a single patient that received GNE gene therapy with lipoplex. And then, you know, the trials that you're aware of. And here I am just including US trials that have been published. So the phase 2 sialic acid trial, the phase 1 trial for MANAC, and the phase 3 of sialic acid. And there are the publications if you're interested in reading them. And the main thing about moving a therapy into clinical trials after you've gone through the process that we've described is that the ability to detect the treatment effect in GNE myopathy is complicated because the disease is rare, so there are not a lot of patients. It's slowly progressive, and it involves different muscles at different stages of the disease. So and therefore you need to power trials that will be able to recruit the number of patients that are available and that will be able to show efficacy in patients in a wide range of the disease. And this is important for natural history studies to be conducted, that were conducted. And the main reason was to understand the disease progression, because understanding the disease progression as measured by different clinical outcome measures is critical for a successful trial design. And I'm gonna try to explain to you why this is. On this paper that was published last year, here we show the decline of, as measured by a variety of different endpoints. So for example, upper extremity strength or lower extremity strength, or the 6-minute walk test, or the IBM-FRS, that it's a type of patient-reported outcome. And why is this important? Because based on the decline, you can calculate how many patients you need in a trial if you know more or less what your expected effect might be, if you know what clinical outcome measure you're going to use to measure treatment effect, and the overall design of the trial. So this is an example. Here I am showing an example for the 6-minute walk test, how we calculate the number of patients needed for a 2-year trial. Let's decide, we decide it's going to be 2 years, but it could be something else. And to have 80% power, so that's 80% chance of identifying treatment effect if the therapy is in fact effective. And so based on these calculations, with all the different numbers that are provided in this table in the paper, you could basically calculate the number of patients that you need for a particular clinical trial design. And that's how you calculate power and use different combinations until you find the power that you think it's going to be appropriate for the trial. And so the calculations on the right are done with natural history data that was collected until this year. So they are slightly different from the calculations that we had back when the paper was published, but they're pretty close. So, and as time goes on and more natural history is collected, these may become a little more refined, but I think we already have a pretty good idea of how to make these power calculations for clinical trials. So based on this information, you could calculate the power, in this case, when you use upper extremity strength with the 6-minute walk test, your power based on these numbers is estimated at a 20%. So a 20% chance of identifying a treatment effect, an 85% chance of not identifying the treatment effect, which suggests that either a longer trial might be needed for this particular endpoint. And so once that is established in a clinical trial, the design of the trial, the other important thing is to make sure you are including patients that truly have GNE myopathy. Because a gene therapy with a GNE gene is only going to help patients that have GNE myopathy. And with recent access to exome sequencing and whole genome sequencing, these identify changes in a variety of genes that may be due to a disease, but that also may be due to the normal variation among humans. And so it is critical that we are able to differentiate those changes in the gene that are truly disease causative versus those that are just a normal variation and that do not lead to disease. This is why the recent American College of Medical Genetics guidelines have said specifically how to identify those variants. And several patients have received a genetic testing report that says something like a variant of uncertain significance, especially when they are getting an exome or a whole genome sequencing. That means that that particular change has never been described and that the laboratory is unclear of whether that causes disease or not. So it's important that patients that are enrolled in trials we have figured out whether their specific changes are truly pathogenic or not. And it typically can be very simple. If you get a genetic testing that says positive, you have 2 pathogenic variants in the GNE gene. But if you have a variant of unknown significance, that's not useful, and it is up to us, the scientists, to determine by a variety of methods whether that causes disease or not. And you need to have 2 affected copies that show that they're pathogenic. And so the one on the bottom that shows only 1 change, that would also not be eligibility criteria. So those are the type of things that we can do as we get ready towards clinical trials. And I want to show you this, and it's meant to be funny, but I wanted to just summarize our story of GNE myopathy so far has shown significant achievements from the scientific community, especially being a rare disease. They're truly outstanding, but it also features a sense of frustration in the face of a lack of an approved therapy, and also courage and determination on the part of the patients and the family members. Truly, it is the patients and their families that inspire us, the doctors, the scientists, NDF, to work together to find a better cure. And so I hope that we all, the patients, doctors, scientists, all the patient organizations around the world, continue to work together. Because as I said before in other occasions, together we are stronger and we have a better chance of getting to therapy by being together. And so I ask for all of your continued efforts. I ask that you have hope, and I ask that you help in any way those that are around you as we continue to be devoted towards this goal. And so finally, my key takeaways are that what I just described. So that scientific advances have been made, and the NDF funded studies are addressing several of the needs that are required to support gene therapy clinical trials. And we are continuing to identify scientific priorities moving forward to facilitate the development of the gene. And now I would be happy to take any questions.
Hi, Nuria. Thank you for that wonderful, elegant presentation with incredibly comprehensive and excellent graphics. For those of you who don't know, Dr. Nuria Carrillo is also a whiz with graphics. So it was really, really informative, even for those of us who have heard, all of these stories. It was really pulled a lot of things together for us. Thank you so much. For that. I had a question about the variants of unknown significance, or VUS, as we see a lot in our whole genomic sequencing studies. What are people supposed to do with that information? Can you give those of us who have seen some of these reports and patients who are told that they have those, what should they be doing, if anything, to try and understand it? What could we be doing as NDF? How can we be directing those so that people aren't left with these giant question marks?
So the American College of Medical Genetics has put forward very specific guidelines of how to handle a variant of unknown significance. This is typically better done by a geneticist. So geneticists are better suited than other specialties at both providing genetic testing results explanations, especially when these results are unclear. And there, a variety of things that may be done include perform carrier testing in the parents to show that whatever new variants are being discovered come from each one of all of the parents. They can also show there are computer databases that you can go and predict how detrimental that particular change may be, and there are a variety of them, and so they are used to determining whether that's differentiating a benign from a pathological mutation. And then in some rare cases, you may need to perform some lab testing in a lab to show that that particular change is disease causing. But in a patient that has parents that are both carriers, that the computational programs think they are pathogenic and that has characteristics of the disease, that should be enough. This is one of the few cases in which muscle biopsy might be useful, because if you have a patient that presented in their 20s and they presented with the typical characteristics of the disease, and you find ring vacuoles on the muscle biopsy and the parents are carriers, I think that builds up enough evidence to then characterize that variant as likely pathogenic. And then what a geneticist would do is they would submit it to the database so that people around the world that see the same already know that that has been established for that particular change.
That's interesting. So maybe what we can do in our scientific advisory committee, now that you're going to be chairing it moving forward, we can talk about what to do as a secondary run with people who do demonstrate these VUS, or variants of unknown significance, and maybe we could ask for muscle biopsies. Let's discuss that, because if that gives us more information, that's just ours for the asking. I think that would be helpful, at least if nothing else, to the patients themselves who have been told that they have those, and I think to the general scientific community. We have some questions, and I want to encourage those of you who have questions to please draft them instead of texting me directly. Please draft them here in the chat box so that I can make sure I see those. Tara Vogel has a question. She wants to know if sialylactose would be a better substrate.
I think the mouse models, there have been studies, so sialylactose, sialic acid, and MANAC were compared next to each other. In the mouse model that I showed, they all really showed pretty similar results. I know that Dr. Nishino has performed more studies, but other than that mouse study, they have never been compared again side by side. And so the answer is, in the mouse model, they seem they're comparable, but we do not, the clinical trials and the studies that have been done have not been harmonized enough for us to be able to compare the results, and I think the sialylactose human experience is limited. So I don't know. I would be interested to find out, but I don't know.
Interesting. All right. And then she follows up with another question, wanting to know, what if someone has one GNE mutation and then one novel mutation?
So that also helps you reaching the let's say, we very rarely see patients with two variants of unknown significance. That kind of lowers my you know, my level of suspicion. When you see a patient that has one variant that has established to be pathogenic and the other is of unknown significance, then that's when you follow the process that I just described. And doing that with two variants of unknown significance becomes a challenge in one patient. So yeah, the two variants of unknown significance in the single patient may be more complicated and less likely to yield a pathogenic change for that.
We have a comment and question from Mangesh. Wants to say that it was a great presentation, compliments you on that. He wants to know, are the toxicology studies from a different preclinical study, but with the same vector, like the RH74, and a promoter, the MCK, reusable for a GNEM trial?
I think that if you're using the same vector, the same promoter, and the same doses, which may or may not be available, I think you should definitely reference them on the IND, and at least it makes your life a little easier when moving forward with the toxicology studies. But you know, I'm not an expert of, and we should probably ask this of a toxicologist, but I have a feeling that you may need to do this with the particular gene as well. Just because the effects of the gene itself, introducing them in cells, you know, do vary from gene to gene and how much they are expressed. And so I do believe a portion of those toxicology studies will need to include the GNE gene as well.
Okay, well, that's good to know. Wants to also know, follow-up question is, does the absence of a viable and reproducible mouse model block the preclinical stages of gene therapy development? The follow-up question is, it appears that this didn't block the clinical trials for, the Sialic and Manac trials. So
Yes.
what's the deal with that? Can you, because that's honestly a question and a comment that we get often. And
Right.
so if you could clarify that for us, you know, is it possible to go forward without one?
Well, so the reproducibility of a mouse decreases over time, because as you breed the mice over and over again, like I was describing, their reproducibility may be lost over time. At the time when those preclinical studies were done for Manac and sialic acid, those were good enough as preclinical, establishing the preclinical for those compounds for us to go into clinical trials. There was also the testing, even though it wasn't biologically relevant, on the M743 mouse. But so, which is why I said that not having a mouse model right now is not a problem we cannot solve. There are multiple ways to solve that problem. You can either find out what's causing the other mouse model to not be reproducible, and that's done frequently in places, in a variety of places, and so identify the cause and fix it. Or we also have these great new technologies to developing mice, which are actually really powerful and are at our disposal. And so I think that just like discovering the gene, it started this process of multiple discoveries that followed. I think having right now a mouse model that works, whether it is solving the old one or a new one, will empower the scientist, and it would make everything move faster.
And I mean, there's a lot of talk from Dr. Marianne Hoising, for example, how I forget her last name, May, the scientist over at NIH that worked with Dr. Nishino on the mouse model, is in fact able to replicate that. I know that Drs. Leck have offered to do that in their lab. And if we do have that and Dr. Martin's or Dr. Rosenbaum's model, that will just strengthen our collective understanding. Is that correct? I mean, there's not going to be a problem if there's two working.
Correct. It is my understanding that at the NIH, they're still able to replicate that same mouse model. I have also heard from other rare disease foundations, what they do is they keep, they have a gene, a mouse genetics company maintain the health and genetic integrity of their mice, so that when researcher needs it, they can have a fresh new breed to use at, you know, at a time. And so once we have figured this out and we have a working mouse, I think that that probably is a good resource to look into, just because it makes the scientists move faster, not needing to wait, you know, for the 6 months until you have a mouse that's working, things like that.
Yeah, the, doctors Leck from Yale have offered to do that and have suggested that in the past. So I think that's something we should probably look into. Everyone agrees that that was a good question on the mouse question. So thank you for that, Mangesh. Nitin wants to know, are we giving up on manic and moving to gene therapy? And I know that, let me just say this, that for those of you who have the same question, I get that question all the time. Let me just explain two things before Dr. Nuria is put on the spot. Number one, doctor, first of all, the answer is emphatically no. We're not giving up on any potential treatment. And MANAC is something that Dr. Nuria Carrillo left NIH in very good shape, and absolutely we're not giving up on it. My understanding is, and the reason I'm answering this question is because Dr. Carrillo has a confidentiality agreement with NIH that we want to honor and not put her on the spot and ask questions about manic. So I will try my best to answer it. And then Nuria, if there's anything you can add to that, please do. We're not giving up on manic. Manic is very much in progress. It is like everything else, COVID that has delayed the bringing together of the patients, the travel restrictions, and the safety of COVID that have been imposed by COVID, that has simply delayed it. But everything is in the hands of NIH and Neuronext and Leadiant, and everything is, to my understanding, ready to go as soon as it's safe to continue bringing patients in. Dr. Carrillo, is there anything you'd like to add to that?
That's accurate. That's entirely accurate.
So no, so I hope that answers a lot of, because that was one of the first things that we heard. Thank you, Mona, for thanking everybody for clarifying that, because that is a question we get very, very often. And the first thing everybody thinks is, oh my gosh, Nuria must be giving up on manic. Not at all the case. She was very, very clear with me when we even began the conversations that she had done everything she could, and now it was in the hands of others to take to phase 2, 3, and finish off phase 2, go into phase 3 if necessary, and then commercialize it. So everything is, as far as we know, on. And I'll tell you what, as soon as we know, we will blast an announcement to you guys saying that they're ready to take patients and so on and so forth. So that is a very, very important point that I appreciate you allowing us to make, Nuria. And for the record, from now on, if you guys do have questions about MANAC, please email us at info@curehibm.org, and we will put you in touch with the right people, either at Neuronext or NIH. So we'll be happy to do that. Another question is, can you talk a little bit more about the biostronic gene therapy? I don't know. I don't know what that means. Has a myostatin inhibitor ever tried in combination with either sialic acid or MANAC? These are very, very educated questions, but I'm not sure I understand.
A lot. I do not know that has ever been done together with sialic acid and MANAC. Right. This was something that was mentioned by Dr. Martin during his presentation. Yeah. In great detail, because that's one of the strategies he's thinking about. It's towards the end of his discussion. He's very well versed on this, so I would certainly guide you to see that part of his presentation, which I know is still available on the website, and look at that. But to my knowledge, it has never been tried together with the other therapies.
Okay, all right. Well, thank you for clarifying that. we have got, I think, dwindling questions. People are emailing me and texting me privately. I just want to clarify to everyone that some of the references that Dr. Carrillo has made to other videos live on our website, are a major part of our education program under the Symposium Speaker Series. Please go to curehibm.org, click on the front page that tells you to take you to our video library. Very painstakingly goes through every single study that has ever been done for GNE myopathy in recent history, explains it in detail, and if you don't have time to look at it, gives you an overview slide and a key takeaway slide. But you'll be able to catch up on every question you ever had about GNE myopathy, including things that are not science-based, but are mostly really just for the improvement of the quality of lives of patients living with GNE, such things as intimacy, practical issues, assistive devices. So please do look through that, because it's a wealth of resource for anyone interested in this disease. Dr. Carrillo, I'm going to just finally lead with one final question. If there's an overview you can give us of the journey that still remains ahead of us and identifying any gaps that we have. Could you reiterate for our donors, our patients, and their families, because I think it's all important, that what is your take on our approach to the IND, our approach to our, like I said, this journey towards gene therapy, because people think of gene therapy as kind of an occasion in which they get, dosed through a needle and then it's gene therapy. But can you explain maybe, or summarize for everyone in this journey, have we been doing it in a way that, because we have no ego in this, but this is all based on the consensus of a lot of scientists, are we taking the right approach? Are there any gaps that we need to identify? And I'm certain they are. What are those? And because I know this question comes up often, how long do you think we're looking at? But could you summarize that for us before we let you go?
Yes, of course. So I think the most important approach of all is to do this together as a community. And I think NDF has certainly been doing a great job of bringing both the patient and scientific community together. And this is, I think, one of the most important and critical factors in moving forward with any clinical development program. In terms of the gaps, I will summarize based on my discussion that I had. Having a mouse model that is biologically relevant, amenable to testing, and reproducible is important to do the preclinical efficacy testing. Having developed and improved scientific tools such as antibodies, enzyme assays, and developing standard procedures for each one of them so that experiments can be replicated across laboratories. So overall, develop a research toolbox for GNE myopathy that can be accessed by any scientist interested in testing therapies for GNE myopathy. And specifically for gene therapy, we need a careful selection of the gene therapy strategy, the vector and the promoter, and with, like any other development program, showing that a strategy is safe and effective, and performing toxicology studies is what allows an IND to be allowed to move forward to early testing in clinical trials.
Thank you. Thank you for that clarification. That was actually really good soundbite for us to have. Anna Warner says, it's so good to see you again, Dr. Carrillo. My heart sunk when I started reading your email that you were leaving NIH. Then, as I kept reading, I was beyond thankful you are now part of this. And I think by this she means NDF. When the trial does start up again, will it be at NIH, she asks?
So for which particular trial, the MANAC trial or the gene?
I think she means the MANAC trial. Please clarify, Anna, if that's not, she says yes.
For the MANAC trial, there are several centers across the US that have been selected for the trial, so it would be multi-center. And I would refer you to clinicaltrials.gov for the multi-center trial of MANAC for GNE myopathy, where you could see where those sites are. And any questions that you may have, maybe given to Lale again, or directly to Dr. Gall at the NIH, who is now in charge of the program there.
Dr. Gall, an old friend. So thank you again, Nuria. Amazing presentation. Thank you, everybody, for joining us. We have a couple more comments. Mona Patel says she echoes Tara's sentiments. I feel so grateful that we all of you working tremendously hard to support us through our journey and working tirelessly to find a treatment. Thank you again, everyone, and we look forward to hearing your feedback on our symposia speaker series. I hope you found them helpful, and we are here if you need us. Please reach out to info@curehibm.org, and we will make sure that your email gets to the appropriate person. Thank you, everyone, and have a good rest of your day. Talk to you soon. Bye-bye.